Okay friends, so in this video tutorial we will going to talk about the drug resistance in very details. Okay, so let's start with the definition. So what do you mean by drug resistance? So drug resistance is a adaptive response of microorganism in which bacteria begins to tolerate an amount of drug, okay, that would ordinarily be inhibitory. That means, suppose let's consider a bacteria named A, and previously it was susceptible to antibiotic X. Okay, now suppose you are a doctor and there is a patient in your chamber who have developed infection of A bacteria. Okay, now you know that yes, X can work well against the A antibiotic, so okay, we will going to use X. You prescribed the X antibiotic in your prescription. Okay, now after some day that particular victim returns and told you that the symptoms of the disease persist. Okay, so under such circumstances, now you try to think about this, that what is the reason why X is not working? Am I made a wrong diagnosis, or there is a problem in the testing? What is this? So upon investigation you find out that no, the diagnosis is absolutely well. Okay, and you have also diagnosed the symptoms well. That means it is confirmed that the bacteria is A. Then why X is not working? So let me explain and let me introduce a new type of threat to our world, that is the drug resistance. Okay, that is the situation in which the bacteria develop resistance towards that type of drugs which was previously very useful against them. Okay, that means now you cannot use X against A. Okay, so that is a simple explanation of what is drug resistance. So now let's move. So another type of explanation I can give. Suppose let's say you are weak in physics. Okay, now you get a good teacher, absolutely a very good teacher, and he explains the concepts of physics very well in a familiar way that you can connect, and after 7 to 8 months you become master in physics. Okay, that means you have developed resistance to physics. Okay, that means now physics is not a threat for you no longer. Okay, now I think it is very clear to you what I mean to say. Okay, so now, so how bacteria develop such kind of mechanism to inactive those kind of antimicrobial drugs? So remember, these things are acquired by bacteria due to the genetic versatility and their mentality to adopt. Okay, that is the reason why bacteria can develop drug resistance. Okay, and if you want to talk about some exciting drug resistant bacteria, so I should say that Staphylococcus aureus is expert in acquiring drug resistance. You get MRSA, you get VRSA, you get VISA, all are types of drug resistant Staphylococcus aureus. Okay, we have talked about MRSA, VRSA, VISA in very details in Staphylococcus aureus video lecture series. You can consult those videos for more better understanding. So now, drug resistance can be divided into two parts. One is intrinsic drug resistance, another one is acquired drug resistance. So let's talk about the intrinsic first. So intrinsic drug resistance can be explained by a simple concept that bacteria must be resistant to those antibiotics that is produced by that bacteria itself. Okay, that means let's say a bacteria produces X antibiotic. Okay, that simply means X is resistant, a bacteria is resistant to X. Okay, why it is? Because remember, what is antibiotic? Antibiotic is the mechanism that produced by bacteria to ensure survival, right? They releases antibiotic to either disrupt the metabolism of other competitors which are present, or to destroy those competitors, right? So why bacteria will produce some type of things that may be poisonous to itself, right? It does not make sense, right? That is the reason it is said that bacteria is resistant to those antibiotics which are produced by itself. Okay, and this kind of resistance is known as intrinsic drug resistance. Okay, and these things are not very much important point here. Okay, we don't need to bother about this because it is limited to a small group. Okay, and it does not affect in antimicrobial chemotherapy. Okay, but our main problem is the acquired drug resistance. Okay, that is, suppose let's go back to that example. A bacteria now develops resistance to X. Okay, but in earlier times X was a very important drug for treatment against A. That means A have acquired the capability to protect itself against X, and this kind of resistance is known as acquired drug resistance. Okay, this is a very important thing to understand, and this is the main concern on the drug resistance. Okay, so we have talked about that drug resistance can be divided into two parts. One is intrinsic. Intrinsic drug resistance simply wants to say us that the antibiotics which are produced by bacteria do not cause any harm to that antibiotic. Okay, and it is not an important concern for us, and it does not affect the antimicrobial chemotherapy, right? Because remember that yes, A produce X, right? So I think I should use different notation, then otherwise it will be very difficult for you to understand. Let's say B, so B produce Y. Okay, that simply means Y does not cause any harm to B. Okay, that does not means that there is another type of antibiotic P which is produced by, let's say, R bacteria, cannot cause any harm to B. It may, right? And that is the reason why we can use P to destroy B. Okay, that means intrinsic resistance is not a problem, but acquired resistance is a problem. Okay, because it is the adaptability of the bacteria to develop resistance against certain antimicrobial drugs, antibiotics. Okay, now let's talk about what type of mechanism bacteria adapt that provides them the property called drug resistance. So first thing is the drug inactivation mechanism. Before going to start that part of the lecture, I want to recommend you to please go and watch the mechanism of antibacterial drugs that I have explained in several videos. There is a cell wall synthesis inhibitor video, cell membrane synthesis inhibitor video, protein synthesis inhibitor video, nucleic acid synthesis inhibitor video, because if the mode of action is very clear to you, then it will be easy for you to understand. Okay, so first thing is the drug inactivation mechanism. So how it is achieved? So first thing I want to mention that in that type of mechanism, bacteria start to produce specific type of enzyme that create a permanent change in the structure of the antibiotic. Okay, and as a result, that particular type of antibiotic or drug becomes permanently inactive against that particular bacteria. Okay, let me explain. So in cell wall synthesis inhibitor video, I have told you that most of the gram negative bacteria have the ability to produce a special type of enzyme named beta lactamase. Okay, and this beta lactamase will destroy the beta lactam ring of the penicillin G antibiotic, and as a result penicillin G antibiotic becomes inactive. Okay, and under such circumstances we should use clavulanic acid, which is a beta lactamase inhibitor. Okay, that is the reason. So here beta lactamase is the enzyme that is produced by the bacteria that ensures the drug resistance, right? Similarly, chloramphenicol acetyl transferase, this is another type of enzyme that is produced by some bacteria. This chloramphenicol acetyl transferase attach a acetyl group to the hydrophilic group of the chloramphenicol, okay, and inactivate it. Clear? So that is how in that particular mechanism they are producing specific type of enzyme that create a permanent change in the structure of the drug and inactivating it. Now let's talk about decreased drug permeability. Okay, in that type of mechanism some bacteria decreases the chance of the drug to enter. Okay, for example, let's say gram negative bacteria. Gram negative bacteria have outer membrane extra, okay, that provide them a natural resistance to penicillin G. Okay, because penicillin G have to bind with penicillin binding protein to work. Okay, and outer membrane prevent penicillin from entering the cell. Okay, so that is a natural type of resistance. Okay, now let's talk about another thing, that is drug elimination or efflux. Okay, so let's consider this is a bacteria. Okay, now these bacteria have some enzyme who named. So now this, in that type of mechanism the bacteria have the ability to produce a special type of protein called, let me erase some part of it, plasma membrane translocase. Okay, so this plasma membrane translocase act as a efflux pump. Let's say this is the plasma membrane translocase, and it will remove all the drug that have entered inside the cell. Okay, and as a result those drugs cannot work. So those bacteria who have the ability to produce this translocase type plasma membrane protein should develop resistance against drug, right? So that is the drug elimination or efflux, and this type of plasma membrane translocase are known as efflux pump that have the ability to expel all the drug that have entered inside the cell. So one type of drug resistance is gram positive and gram negative bacteria have developed resistance against tetracycline because of the overproduction of the translocase related plasma membrane protein that act as a efflux pump. Okay, so we have talked about that bacteria have developed mechanisms that have the ability to produce some specific enzyme that will create a permanent change in the structure of the drug, and as a result they become inactivate, right? And second one is, in that thing we have talked about that bacteria have decreased the permeability of certain antibiotics, right? For example, the penicillin G in gram negative bacteria, and in the above mechanism I have told you about the efflux pump, right? Some bacteria have the ability to produce the plasma membrane translocase protein. Okay, this translocase protein will allow bacteria to expel all the drug that enters inside the bacteria. Okay, now the fourth point is change of drug receptors. So remember, antibiotics or any kind of antimicrobial drugs are target specific. Okay, if you take fluoroquinolones, it will target DNA. If you take rifampin, it will inhibit RNA synthesis, right? If you talk about chloramphenicol, it will target protein synthesis, right? That means their target is very specific. So bacteria also have developed a mechanism that will change their specific target. Okay. For example, the affinity of the ribosome for erythromycin and chloramphenicol is decreased by changing 23S rRNA, okay, to which they are bind. Okay, so that is the fourth type of mechanism that bacteria can acquire. And the last one is the change in metabolic process. Okay, some anti-metabolites who especially targets various steps of important metabolic pathway may also not work. Under such circumstances, you should think about this, that bacteria may have developed other pathway to complete the need, okay, or it uses the ready-made product for that particular purpose. For example, let's take a normal example. Suppose you ask your mom to cook chow mein. Okay, now your mom said that she is today not feeling well, so she cannot cook chow mein for you. Okay, under such circumstances, your desire to eat chow mein will be hampered. But under such circumstances, you have special type of apps in your phone who can deliver you ready-made chow mein from restaurants within 5 minutes. Okay, and you ordered in the app and you get your chow mein. That is the thing. Suppose bacteria is performing a particular type of metabolic pathway so that it will get the end product that will be useful for bacterial own metabolism. Okay, now you have incorporated some drugs here to inhibit the metabolism, okay, inhibit that particular metabolic pathway. So as a result, the product will be not produced, okay, and as bacteria will not get this, bacteria cannot survive. But you observed that bacteria still survive. So here the reason is bacteria have developed a alternative way to get that product. It is now not relying on this pathway only. Okay, that is the thing. That is the number 5 mechanism. Okay, so certain drugs that have developed resistance against sulfonamide because they have the ability to take preformed folic acid from their surrounding rather than synthesize it themselves. Okay, so we have talked about sulfonamide. They are folic acid pathway inhibitors, right? And folic acid pathway gives us nucleotides, right? So bacteria have developed mechanism that allows bacteria to use preformed folic acid from the surrounding environment. So why need to go and use its own energy to perform another pathway, right? And that is the type 5 mechanism. So that is the 5 mechanism you need to remember from drug resistance. Okay, now how those type of resistance is developed. So remember, if two things occurred with bacteria, then only bacteria have developed resistance. One is spontaneous mutation in chromosomal genes, and the second one is acquisition of new gene or set of new genes. Okay, that via transfer mechanisms from another species. If any one of the two situation occurs with bacteria, then bacteria develop resistance. Okay, so spontaneous mutation. So if you have studied about bacterial genetics, then I think you know that bacterial genomic content undergo spontaneous random mutation, right? And this spontaneous random mutation can change the genetic content, and as the genomic content is changed, then the protein produced by itself will also be changed, right? But this type of spontaneous mutation is not a too big concern for us, because the end result will create a slight change in microbial sensitivity, and that can be easily covered by using a larger dose of the drug. But our main concern is plasmid mediated resistance. Okay, suppose bacteria have acquired a resistance gene by acquiring a plasmid. Okay, that plasmid can be acquired by bacteria by various type of processes, okay, such as conjugation, transformation, or transduction. Okay, now as this particular type of resistance plasmid is inside the bacteria, so now when you will try to kill the bacteria by using the antibiotic whose resistance gene is present inside that plasmid, then the bacteria will not die, right? Because it has the resistance gene to fight against that particular antibiotic. Okay, so that is the concept of plasmid mediated resistance. Okay, so not only that this plasmid that bacteria have acquired will provide resistance to that bacteria, but also when that bacteria undergoes cell division, this plasmid will be transferred to its upcoming generation. Okay, and as a result, the upcoming generation will also get resistance against that particular type of drug. Okay, that is the concept. Okay, and interestingly, this type of things occurs naturally even before the bacteria is exposed to the drug. Okay, such traits are lying in wait for an opportunity to be expressed and to confer adaptability on the species. Okay, bacteria may also develop resistance due to some transposome or transposable elements. So transposable elements are what? They are jumping genes. They can be copied and inserted from one plasmid to another plasmid, or from one plasmid to another chromosome. Okay, and due to this insertion of transposable element, bacteria may develop resistance. Okay, and this is the hereditary resistance. I have told you that during cell division, the plasmid that the bacteria have acquired may be transferred from one generation to another generation, and as a result, those generation will also get resistance towards that particular type of antibiotic. Okay, so that is the fourth type of situation. If it is occurred with some bacteria, then it will develop resistance. Okay, so in that video we have, let's take a quick recap what things we have seen in that video. So drug resistance are of two types. One is intrinsic drug resistance, another one is acquired drug resistance. So intrinsic drug resistance is the normal thing, we don't need to bother about that. It is the resistance of the bacteria that it has developed towards the antibiotic that is produced by itself. Okay, this is the normal thing, but our main concern is the acquired resistance. Okay, then we have talked about the 5 mode of action that bacteria uses to protect itself from drug. One is it may produce some enzyme that will inactivate the drug permanently. Second one is it may change the target of the drug. Third one is it may decreases the permeability of the drug. Fourth one is it may produce some efflux pump, that is plasma membrane translocase. The fifth one is it may use some another metabolic pathway or use the product ready-madely without going the pathway where we have set the inhibitors, and as a result those anti-metabolites are not working. Okay, and we have also developed we have also discussed how those bacteria get those kind of resistance. One reason is spontaneous mutation, second and most important reason is plasmid mediated resistance, third one is transposable mediated resistance, and the fourth one is hereditary resistance. So this is the fourth type of way how bacteria can develop resistance, and that is the end of that video lecture. So I hope this video will be very helpful for you to understand the concept of the drug resistance. So thank you friends, and take care.